our project is molecular TDA, building models to personalize drugs. We're all from the Jahanakya lab, and my name is Divya, and I'm presenting with Pransh and Minjee. Next slide, please. Okay. Personalized medicine is changing the way we treat disease. Instead of a one-size-fits-all approach, it helps patients receive care that is tailored specifically to them. This not only increases the chance of successful treatment, but also helps reduce side effects and makes drugs more effective at treating symptoms. Our goal is to take this idea further. We're building a model that analyzes a dataset of 7 neurodegenerative diseases and the drugs used to treat them. By using topological data analysis, or TDA, we can uncover hidden patterns and similarities between how different drugs affect these diseases. With this approach, we hope to improve how we match patients with the right treatments, bringing us closer to truly personalized medicine. Next slide. Next, a little bit about the drug discovery process. The drug discovery process has 5 main stages, as shown on the screen. The stages are discovery, preclinical trials, clinical trials, FDA review, and manufacturing. The first stage is discovery. In this stage, drug targets are identified and compounds are tested. This is the first stage of the drug discovery process. After this, the selected compounds will go to preclinical trials, which is where lab and animal products are used to test these compounds for safety. If the compound passes that stage, it'll go to the clinical trial stage, which is human testing. In the clinical trial stage, the stage has 3 stages, phases 1, 2, and 3. In each of the phases, the population of people that the drug is getting tested on increases. If the drug passes stage 3, it'll move on to stage 4 or FDA review. In this stage, it is made sure that the compound passes all the FDA rules and compliances. Finally, the product moves on to the manufacturing stage. In this stage, the product is scaled up and the production is marketed. Drug development is a costly process based on this diagram. In fact, 90% of drugs don't make it to the clinical trial stage, and it costs between $314 million and $4.6 billion to develop a drug. As a result, the drug discovery industry is inefficient and has room for improvement. Next slide. So why our diseases? Our project focuses on 7 neurodegenerative diseases, namely Alzheimer's, Parkinson's, FFA prions, hidradenitis suppurativa, or HS, amyotrophic lateral sclerosis, or ALS, and Huntington's. So why did we choose these 7 neurodegenerative diseases? Despite their differences, they all share key features, things like protein agitation, neural loss, and mitochondrial dysfunction. These commonalities make it possible to compare them meaningfully while still capturing a wide range of biological variation. We chose a diverse spectrum, both well-known diseases like Alzheimer's and prions, and rare ones like Huntington's because each offers a unique insight. And critically, none of them have a cure. This makes drug discovery in this space not just important, but necessary. On top of that, the molecular and genetic heterogeneity across these diseases gives us a rich, high-dimensional dataset, perfect for using techniques like dimensionality reduction and topological data analysis. This allows us to uncover structure in the data and explore drug similarities that might otherwise be hidden. As you can see on the graph on the right side of the screen, the number of deaths from Alzheimer's per 100,000 people in the United States is really high and increasing over time. This has to be fixed. Next slide. To better understand high-dimensional data, we use several dimensionality reduction techniques. 3 common ones that we are using in our project are UMAP, or Uniform Manifold Approximation and Projection, TSNE, or T-Distributed Scholastic Neighborhood Embedding, and PCA, Principal Component Analysis. Each of these dimensionality reduction techniques has its strengths. UMAP is known for preserving both local and global structures by using manifold learning. It gives us a balanced, meaningful representation of our data. TSNE focuses on preserving local relationships, meaning it captures structure as well, it captures cluster as well, but often loses the big picture on how these clusters relate to one another. PCA, on the other hand, uses variance. It keeps the directions while the data varies the most and discards the rest. It's fast and linear, but it doesn't always capture the underlying structure as clearly as UMAP or TSNE. These three tools help us simplify complex data sets while keeping the most important information intact. Next slide, please. Machine learning, or ML, is all about teaching computers to recognize patterns without being explicitly programmed. Instead of hard coding rules, we let models learn from data. There are two main types of machine learning: unsupervised and supervised algorithms. Unsupervised algorithms work with unlabeled data. They find patterns, groupings, and structures without any human guidance. This makes them great for clustering, anomaly detection, finding associations, and even dimensionality reduction. Supervised algorithms, on the other hand, are trained on labeled data. The model learns from examples where both the input and correct outputs are shown. Over time, it builds a function that can make predictions on new, unseen data. Both of these approaches have their strengths, and choosing between them depends on the kind of insight we want from the data.
So specifically in our project, we investigated a variety of machine learning techniques. First, we looked at neural networks, which are computational models that are inspired by biological neural systems. They have multiple layers of interconnected nodes or neurons that process information through weighted connections. Specifically, we examined autoencoders, which are subtypes of neural networks used in unsupervised settings for dimensionality reduction. So they learn compressed representations of the data by minimizing the reconstruction error, and that allows us to keep the key structural features, but in fewer dimensions. Next, we also looked at k-nearest neighbors, or KNN. So this is a supervised classification method that assigns labels based on the majority of the k-nearest data points in a feature space. It's intuitive and effective when clear separations exist among the different classes. And then finally, we explored k-means clustering. This is an unsupervised technique that divides the data into a k amount of clusters by minimizing the variance within each cluster. It iteratively updates centroids and reassigns points to form groupings. And this method was used to explore whether natural clusters in the data aligned with known drug classifications. So these algorithms overall provided us with insights into the structure and relationships within our data set and helped us with later analysis. Next slide. For our methodology, it evolved many stages. We first began by investigating public data sets such as DrugBank and PubChem to identify drugs that were relevant to our 7 neurodegenerative diseases. And we collected associated molecular properties and gene targets. We also conducted a review of recent applications of topological data analysis and machine learning in biomedical research specifically to help us with our methodology. But we used both R and Python for data processing, analysis, and visualization. And we used Python libraries such as Pandas, scikit-learn, UMapLearn, and Kepler-Mapper. We cleaned the dataset, standardized features, and then also applied dimensionality reduction techniques to prepare the data for analysis. And then we analyzed the reduced data using persistent homology and clustering models to look at different topological features and statistical structures. For visualizing the data sets, we used barcodes, persistent diagrams, 2D, 3D projections to help highlight similarities between different drug profiles. Next slide. So our data set finally consisted of 210 pharmacological compounds, and each was associated with one or more of our 7 neurodegenerative diseases. Each compound had 73 molecular properties, including structural features such as molecular weight and rotatable bond count. physiochemical descriptors like log P, solubility, and polar surface area, and then biological properties such as blood-brain barrier permeability. And all of these properties were gathered from one of the following publicly available datasets, including DrugBank, Zinc, PubChem, ChemBL, PDB, SwissADME, and PKCSM. And this helped us build our complex dataset, which was the foundation for our project. Next slide. To ensure the integrity of our dataset, we used rigorous data cleaning procedures. First, we made sure that properties with less than 70% of the data across all drug entries were excluded to minimize the impact of missing data on model performance. We also removed redundant features to prevent overfitting and dimensional redundancy. And then we also normalized our features using logarithmic scaling, and numeric features were standardized to consistent units and significant figures to make sure there was a fair comparison. Finally, we converted our qualitative data to quantitative data. For example, descriptors such as low permeability were mapped to numeric scales to allow for quantitative analysis. Next slide. To execute our study, we used a variety of tools across data science, chemoinformatics, and visualization. We used R for initial statistical analysis and exploration of drug property distributions. R's libraries for bioinformatics and data visualization helped us to interpret our data set. Pandas specifically was used for data manipulation and data cleaning. It allowed us to efficiently manage our drug data set, filter and clean features, and prepare the data for our dimensionality reduction. We also used RDKit, which is a chemoinformatics tool, to compute and extract the molecular descriptors and chemical properties from their drug smell structures. And this allowed us to contribute to our data set and help with our analysis. We also use Anaconda as a platform to manage our Python environments and packages, and it made sure that there was compatibility across different libraries such as RDKit, Kepler Mapper, and scikit-learn without dependency errors. Jupyter Notebooks allowed us to have a coding environment for writing and documenting our methodology, as it allowed us to combine code, visualizations, and more into one place, which will help us improve the reproducibility of our study. Python was used as our primary programming language for our study, as we used it to implement the entire machine learning and TDA pipeline, as this language was very efficient, making it ideal for prototyping and analysis. For visualizing our results, we relied on matplotlib, and it allowed us to create clear graphs and plots, including dimensionality reduction projections and heat maps of drug features. And then finally, for TDA specifically, we used Kepler Mapper as our main tool. It allowed us to construct visual network graphs that revealed the high dimensional relationships in our drug data set and find topological features that conventional clustering methods wouldn't be able to find. Next slide. Okay. This slide shows our exploration into different technologies such as R and Python. On the left, we have a heat map of our drug data set, and it's able to capture the relationships between all of our 210 drugs and 73 properties. Each row represents a drug, and each column represents a specific property, such as molecular weight, H-bond donors, or log p-values. And this heat map helped us in many ways. First, it helped us identify clusters of drugs with similar profiles, which were early candidates for exploration. Additionally, it helped us split anomalies, such as drugs with outlier values across multiple properties, and we either investigated those by cross-validating them throughout multiple data sets or excluded them entirely. We also used the heat map to remove properties which did not meet the 70% threshold. And then on the right, we have a reference benchmark of TDA on a breast cancer data set, and this was able to show us how TSNE and UMAP behave on complex biological data sets. And this data set specifically was well studied and had clear separations, so it helped us create expectations for the clustering behavior on our own data set.
These are samples of our UMAP and TSNE reduced visualizations with different iterations testing different parameters for our Kepler mapper visualization. And it was done to identify the ideal parameters for mapping the reduced data after running UMAP and TSNE. On the left is our UMAP visualization testing the number of cubes and the percentage overlap. The number of cubes is shown on the x-axis, and the percent overlap is going to be perk overlap on the y-axis. On the left, our UMAP visualization shows the different iterations of testing the number of cubes and the percentage overlaps, where n cubes is the parameter in Kepler mapper representing how many overlapping intervals or cubes or bins are used when covering the projected data space. Additionally, the perk overlap shows how much the adjacent intervals or cubes overlap with one another when covering the data space, because Kepler mapper slices the data into projection dimensions from like PCA, TSNE, or UMAP into n cubes, which are grid intervals. We can see that the ideal number of cubes and the percentage overlap fall between 10 to 15 cubes and 0.1 to 0.2 perk overlap. For TSNE, the ideal number of cubes appears to be somewhere between 14 and 15 from 0.1 to 0.3 overlap. PCA was another type of dimensionality reduction technique that we did try to use on our dataset. PCA is a linear dimensionality technique that is commonly used for reducing dimensionality in linear datasets. It extracts the most important underlying features in datasets while improving the model performance, and it typically reduces data into 2 or 3 dimensions. However, if too few of the components are retained, then there is a very high likelihood that there will be some information loss. Additionally, PCA assumes that there is a linear relationship between the different variables. And on the right, we see a PCA reduction and visualization of the iris dataset, a dataset that contains data points about the 3 different species of irises, including the petal length, width, sepal width, and length, which are linearly related. Our dataset, however, is nonlinear. For example, the number of atoms and the pKa, or the blood barrier score and hepatotoxicity, aren't really linearly related. And thus, our dataset was not ideal for the PCA reduction trial, and we got inconclusive results. Some limiting factors for our project include incomplete feature coverage, varying data sources where the reported information is conflicting, and the limited number of drugs available on market and the limited amount of information available about the drugs on market, specialized for our neurodegenerative diseases. For example, the drugs we included in the study mostly focus on treating symptoms and slowing down the progression of the disease. So our data would not be too helpful in finding specific cures for our diseases. Second, our current model relies actually only on chemical and pharmacokinetic properties. This means that we looked at factors such as molecular structure, molecular weight. However, we did not include data about the pathway activation and patient-specific variability or gene expression or associated genes. These biological layers could significantly influence how effective a drug might be in real-life scenarios, so the exclusion is a considerable limitation to consider. Third, we found that the dimensionality reduction and topological analysis, which we use to uncover patterns, are highly sensitive to parameter tuning. And for example, parameters such as n cubes and perk overlap in the mapper algorithm can dramatically affect the output. Without careful calibration, the results might not generalize well. So we believe that some of our future work should focus on optimizing these parameters and running stability tests to make sure that these findings are generalizable and interpretable. For our future work, our focus is on refining the entire pipeline from dimensionality reduction to lead identification. We plan to optimize the dimensionality reduction parameters and visualization parameters that feed into topological data analysis. This includes experimenting with settings in UMAP and TSNE as such the previously mentioned and neighbors and minimum distance and perplexity to identify which combinations produce the most informative low-dimensional structures for our data. Next, we will apply unsupervised machine learning models, specifically autoencoders, to learn compressed representation of drug profiles. Autoencoders are well suited for this because they can capture linear relationships and reduce noise, and reveal underlying patterns that may not be more visible through other methods. Using the compressed embeddings, we can perform TDA and find similarities between the properties of drugs, and hopefully we will be able to identify a profile for potential drug leads for the different diseases. In this slide, we highlight how our research can translate into further development and future applications. Integrating TDA into drug discovery, the drug discovery process, the industry can potentially gain several benefits, such as accelerating compound generation by revealing underlying structures in large chemical data sets that traditional methods may overlook, and can potentially explore more meaningful chemical space faster. Second of all, TDA can be a supportive tool for drug repurposing by identifying topological similarities between the previously existing drugs and novel compounds. We could also potentially suggest using TDA and in conjunction to machine learning models to interpret clustering results and predict exact potential drug profiles and specific compounds to potentially make the screening process much more efficient. Thank you for listening, and thank you to our PI, Saharja Hanikia, ASDRP, and the Olive Children Foundations. Our references are in the QR code down in the corner. Do we have any questions?